our bone and cartilage. All right, and why are we paying attention to these two types of connective tissues? That's precisely because the endoskeleton of vertebrates is made up of either of these two, correct? Okay, and we have taken a look at the histology of cartilage last time, and in connection with this, I asked you the question, why is it that there are no cartilaginous tetrapods? I suppose you've answered the question. Yes, and if you have done that, may I get your 1 point? It was supposed to be an assignment, right? Yes, and the others are prepared so that the others are looking at each other. So we have an assignment. Okay. Alright. So for those of you who have an assignment, may I have your 1 form? Okay. And if you will remember what I told you last time, I consider assignments as quizzes, right? So if you don't submit your assignment, you don't have. Alright. So any more 1 forms? Okay, very good. All right, and Johnny, can you give me what is your answer?
The cartilaginous bone is irritated.
Cartilaginous
The
what?
cartilage, like skeleton is irritated by bone for support of the bone.
Okay, for most vertebrates, cartilage is replaced by bone. Okay, but the question is not that. The question is why are there no cartilaginous tetrapods? Meaning, you and I are tetrapods, right? But we prefer for bone instead of cartilage instead. What do you think is going to happen if your cartilage is skeleton? Any volunteer? But just simply give me your answer from your one book. Yes, me? Jenny, right? Is Jenny also? Jeannie, that's Jenny. Oh, okay, the same answer as Jojo. We have bones here, cartilage, aquatic, and collagen. Oh, so the same thing. That doesn't answer the question. Can you imagine, example would be sharks and skates and rays. They are cartilaginous, isn't it? Yes, okay. So they have decided to live on aquatic environments. Everybody knows that. So why not transfer on that? And retain the skeleton as well. Remember what did we say the cartilage is? Flexible, isn't it? Yes? Alright. So can you imagine a tetrapod with a very flexible skeleton walking on land? That would be very difficult, isn't it? Okay, so to walk on land and to support the massive structure of such vertebrates, you have to make sure that your skeleton is going to be rigid instead of flexible. Okay? So there you are. So cartilage could not withstand, right, the compressional forces that our bodies undergo and absorb whenever we walk on land. Okay? So that's why we should know. So I hope that some of you have correct answers. Alright? So last time we also have mentioned that bone actually is what? It's histogenesis. Good morning. It's histogenesis. Do you understand what I'm saying? Histogenesis. Genesis means the beginning, the creation, isn't it? And histo has something to do with tissues. So the creation of the tissue, okay? That's histogenesis. So in the histogenesis of bone, one of the ways by means of which you can do this is actually to lay down, mesenchymatous tissue. Correct? How do we call this mesenchymatous tissue once again? Next letter L. Plasma. Very good. Alright, so plasma is just a mass of mesenchymatous cells that can, of course, differentiate later on. And such differentiation will be under the word of factors that will help this underlying cells go on and become bone cells. Okay? So, these are going to become bone cells indeed because these mesenchymatous cells are going to become what kind of cells? There are 3 kinds of cells: bone cells, ossification. And when we say chondral, what comes to mind is chondrocytes. What are chondrocytes? What are chondrocytes? Osteocytes are bone-forming cells. Chondrocytes are cartilaginous. Very good. Alright, so when we say endochondral, and chondro, something to do with cartilage cells, that means to say that we begin what? We begin forming bone by first laying down cartilage. Alright, and what will happen to cartilage later on is that it will become ossified to form bone. So this is precisely the reason why it is known as endochondral ossification. Alright, and here is an example of such kind of bone that develops in this manner. And by the way, endochondral ossification gives rise to what is known as replacement bone, and of course, you know what is the reason why it's called replacement, right? Yes? And still another way by means of which these bones can be named is for them to be known as cartilage bones, which is how they typically are referred to. Alright? So cartilage bones imply that they start out as cartilage, later on becomes bone. Yes? Yes. Okay, very good. Now before I talk about the method of endochondral ossification, tell me first, why should you start with cartilage? Why not start with the placenta? It's much simpler. Take a look at the example that is shown in the diagram. We start with a leg bone, right? Or any of the long bones of our body will do actually, which tells you that the long bones of our skeletal system arise by means of endochondral ossification. Whereas the membrane bones or the subdermal bones are what? They generally are found on the skull, right? That was what we mentioned last time. So why start with cartilage bones? Why start with cartilage? Take a look at your leg bones or your arm bones. Why should they be cartilaginous at first? Or even portions of our skull are also cartilaginous at the beginning. Not all of the bones in the skull are membrane bones. Some of them are cartilage bones as well. Especially the ones that form the roof of the skull, they are cartilage bones. So why? It should be quite easy. We all start out as small creatures, right? Yes, okay. Who came out of his mother's womb already on this size? Nobody, right? Yes, alright. So, what is the implication of that? That as we mature, as we grow older, yes, you are saying something?
We're constantly growing.
Yes?
We're constantly growing, so our bones are...
Alright, so we grow taller. That demands that our bones should also be able to, what? To supply the demand of that growth, isn't it? Yes, very good. Alright. So, what is the nature of cartilage that makes it a good candidate for such kinds of requirements? They are elastic and flexible. These are characteristics of cartilage. Alright, so this is endochondral ossification. We all first start with a hyaline cartilage model. Okay, and if you will remember last time, there are three kinds of cartilage, right? What are the three kinds again? One is hyaline, the other is fibrocartilage, the third one is elastic cartilage, correct? Right, so what is our model here? Our model is in the form of a hyaline cartilage, alright? And as this progresses, what is going to happen next is you have the formation of a what? Of a bone collar, and you can see the reason why it's known as a collar, any collar for that matter, because it is found here just where you are forming also your periosteum. What is your periosteum? I think we mentioned that last time. The periosteum is the covering of osteum, the covering of bone. In that manner, in a similar manner, what would you call the covering of cartilage? It will be very nucleus, right? Yes? Alright, so the periosteum is the coping of bone. And in this region are going to form the bone collar, and in the middle of this bone collar is going to be found a primary ossification center. Okay, so there you are. So when we say primary ossification center, primary means of course that is the beginning, and when we say ossification, that means to say that this is where bone tissue will begin to form. Understood? Why? Because what we have there initially is cartilage. Right? So, my primary ossification center is going to form. And by the way, just to make sure that we're going to be correct in our terminologies here, remember that the ends of bones are known as, how do we call the ends of bones again? Starts with the letter E, next letter P, epiphysis, alright? And of course, the middle portion of the bone is your diaphysis, correct? Yes, okay, so where do we find the primary ossification center? Where do we find the primary ossification center? Yes, we find it in the diaphysis, alright. Now, as the cartilage here begins to what? Begins to ossify, meaning to say they begin to form into bone, take note that blood vessels are going to invade the bony tissue. Alright? And these blood vessels, as the name suggests, will of course be the very blood vessels that will bring nutrition and all other requirements of our developing bone, isn't it? Yes. And if you are going to remember what the histology of bone is all about, where will these blood vessels spend? Of course, there it is shown as, a major blood vessel that penetrates the ossification center. But having known what is the histology of bone, especially compact bone, last time, where will those branches of the blood vessels go? Into the one. What is the unit structure of bony tissue? I how? Okay, so what is the unit structure of bony tissue? We call that a Haversian system, otherwise known as an ostium, remember? And at the middle of the Haversian system is a cavity. What is that cavity known as? That cavity is known as the Haversian canal, okay, which will connect to the many lacunae present in the lamellae, correct? And are connected to each other by means of small channels called canaliculi. Remember that? Remember? Okay, so what is the nature of this blood vessel now? This major blood vessel is going to send out branches to each and every Haversian system. Clear? Alright? And because it now supplies each and every Haversian system in the bony tissue, it will now be able to send nutrients and other requirements to individual lacunae via your canaliculi. Understood? Are we clear here? Yes. Are you imagining? Yes, of course. Okay, so there you are. So these are now the blood vessels. And of course, not only blood vessels are going to invade the tissue, respectively too, you will find nerve vessels as well. Nerve supplies are the accompaniment of the so-called blood vessels. And you will notice here that ossification continues until this part of the diaphysis is fully converted into bone. Alright? So what remains here are still cartilaginous, isn't it? Okay, so the epiphysis is still cartilaginous. So what do you think is happening here? And this is very visible in the diagram. What is happening to the bone as ossification is taking place? It comes on. It becomes on. It becomes longer, isn't it? Yes. All right. And this will also, of course, answer the demands of the fact that you are now growing taller, isn't it? Okay. Now, as this becomes fully converted to bone, the cartilaginous area of the epiphysis now soon forms what is known as the secondary ossification crystals as well as nerve endings. And eventually, what happens to the centers of ossification? As the bone grows longer and fully ossified, what happens to both the diaphyses as well as the epiphysis? They become converted to bone. Alright? And it's still their cartilage.
Yes.
Yes. Which part?
The jaw.
Where?
The uppermost part.
At the uppermost part. And that uppermost part has an anatomical name. The epiphysis still has cartilage left. Only where? Only where we find articular cartilage. Alright? And that is the cartilage that what? That is the cartilage that faces the other surface of the adjoining bone in the joint. Correct? Alright, so why should we retain cartilage there? Why not really convert it to bone alone? That will be what? That will be difficult because these joints are the very spaces or the very areas where movement takes place. So if you don't put there something which is flexible, this is going to cause the rinagaak of your bones every time you move, isn't it? Yes? Okay, so there you are. Now, in people who suffer from deposits of uric acid among their joints, what happens is, alright, what happens is this becomes very painful because the articular cartilage will no longer provide as much benefit in terms of frictionless surfaces, but the uric acid crystals will cause the tremendous pain which is experienced by these people. Okay, so there you are. So this is what we refer to as endochondral ossification. And take note that this is going to answer, as what we have said a while ago, the requirements of what? Of growth. Okay, so here on this side is an illustration of the two bones in a joint. And one of this is the femur, the second one is of course the tibia. Alright, and as shown, the epiphyseal plates of both are the ones which articulate with each other, and these are the surfaces that we said is still made up of articular cartilage. By the way, what kind of cartilage is it? Is it hyaline? Is it fibrocartilage? Or is it elastic cartilage? It's actually fibrocartilage. Okay, so there you are. That's why when we enumerated last time the sources of these kinds of tissues, it was cited that articular cartilage is fibrocartilage. Alright, now what is happening here is something that we would like to look at in detail. Okay, so once again, this would be the epiphysis, right? And this will be the diaphysis, alright? Where there is your primary ossification center. Okay, now what do you notice here? That a portion of this shall start out as cartilage, later on become ossified, and that would seem to be illustrated here in the form of zones, isn't it? Yes? Do you see a zoning? Do you see a zoning? The purple and the blue, yes. Okay, so we have a zone here that is still made up of what? That is still made up of cartilage, right? Okay? And as we progress downward, what do you notice about the cartilage cells or the chondrocytes? They become what? Yes, ossification. That is precisely the reason why what we're looking at right now is ossification. But what do you notice about the cartilages? First, the chondrocytes are quite small, right? But then, afterwards, they go what? They become aligned, okay, in columns, right? And not only that, what happens to the sizes of this cartilage or this chondrocyte? They become larger. Okay, so there. And after they enlarge, what will happen to them? They are going to degenerate. Okay? And the degeneration of this chondrocytes will lead to calcification of this zone. Alright? And that is the reason why we refer to this as calcified cartilage. And when we say calcified cartilage, is this in any relation to ossification as well? Yes, because what do we mean when we say calcified? Calcium salts are deposited. Alright, so remember, what are the crystals that are found in bone? What is deposited in bone is not pure calcium. What is deposited provisional calcification, right, from the word calcify. And what do you find here? You just deposit calcium salts here, but eventually what will happen is,
They will form.
what will happen is, these chondrocytes are going to degenerate, right? And soon they are going to be replaced by what? By osteoblasts, okay? They will form bone around themselves, and once they are trapped within their own matrix, they become osteocytes, okay? And what you form now around this are going to be what we refer to as endochondral bone, okay? And this is the reason why we call this endochondral ossification. What kind of bone is endochondral bone? This is what we refer to as cartilage bone. Correct? Or initially, as the previous slide suggested, that was what we call replacement bone. Clear, everybody? Okay. So anyway, so this is endochondral ossification, and there is something that I would like to point out as well from this diagram, that the beginning of the formation of any bony tissue is not to become compact bone. Alright? It's not to become compact bone. There are hollow spaces among the osteocytes, correct? And among the bones that are formed by these osteocytes. And so as a result, these spaces eventually are going to become fused. Alright? And when they fuse, this converts the primary bone into what is known now as your secondary bone. Clear? Are you imagining what I'm saying? Alright. First, you form endochondral bone, which have a lot of spaces among them, right? Okay? And then, as the spaces fuse, the bony tissue are going to be forced, alright, into the periphery, and so they become secondary bone. And what do we call primary bone versus secondary bone? Primary bone is many spaces, spongy bone, okay, or cancellous bone, or how did I refer to it right a while ago? Primary bone, okay. Whereas when they are pushed towards the periphery, they become more compact, and so as a result of this, secondary bone is also known as compact bone. This is the bony tissue which is made up of aversion systems. Clear? Okay, what happened to the fused spaces? How do you call the fused spaces? How do you call the initial spaces before? These are what we refer to as trabeculate. Alright, so the trabecular spaces are going to fuse, and this time you have a continuous cavity within the diaphysis of a long bone. How do you call this? I think the word is somewhere here. No, it's not. Okay, but that is what we refer to as the medullary cavity. I think it is in the former slide. Okay, it is in the medullary cavity. Do you see the medullary cavity? Alright, so here. So that's the medullary cavity, but this is better known as the space within the bone that is also known as the what? That is the space or the cavity of the bone marrow. Clear? Okay, so the bone marrow is what? The bone marrow is simply a result of the fusion of the trabecular spaces during endochondral ossification. Clear? Yes? Okay, so if you are asked what is the bone marrow cavity, can you explain that on the basis
Of the endochondral spaces in ossification.
Not quite like you're just mixing up the terms. Okay, so anyway, that's endochondral ossification.
Cycloplasty.
And take a look. Take a look. What do we find here? Alright, we find here an epiphyseal plate, which as you can see is cartilaginous or bony. It's cartilaginous, yes. But do you expect that there will come a time that all of these regions of the bone is going to be bony.
Well, yes, possibly.
Yes, all right, except the one.
The rib.
Except the articular cartilage, right? We said articular cartilages are going to retain that particular composition forever and ever. But we, as what you have said, there will come a time that all of the regions of the bone, the diaphyses, the epiphyses, and the epiphyseal plate will be fully ossified. When is that? Fully ossified. Meaning that everything is bony already. Konti gulang na actually, konti gulang na. But this will come in at a time when you have reached your
Oldest possible age.
And when is that? For ladies, for women, this generally is around 18 years old. Okay? And for the gentlemen, this will be around, on the average, 21 years old. So you are already 18. All of you are already 18. You are not 18, you are 20. Alright, but you're all 18 and above, right? Nobody here is 17. You're only 17. Huh? You're very young. Most of you are 17 first year, right? Yes, alright. But let's say, for example, you are all 18 now. If you look at yourself, that means to say that that will be the maximum height that you will ever attain in your whole lifetime. Okay? If you're only 17, when are you turning 18?
Tomorrow.
Kadali.
Tomorrow.
So remember, that will be your maximum height. I thought you were good. Nina! Alright. Nina, you're not yet 18. So meaning to say you're still going to grow 3 inches taller? You're going to turn 18 when? March pa. Okay, so lawig-lawig pa. Yes. You have a question. Martin, you have how many years to grow? Dali na kayo na super exciting siya na yung question. Imo question natin. Okay. Very good. Okay, can you answer your own question? If bone is resorbed, remember that osteoclast can resorb bone, old bone. So the question of Nina here, which is actually the question of Martin, which he hates right now, to be pointed out as the source of the question. I don't even know that question. Okay, but what happens to the old bone?
Recycled into
What happens is when the osteoclast resorbs, there is a what? There is an enzymatic process that is going to reduce the bony tissue into its simpler components. Remember that bony tissue is collagenous. Yes, it's collagenous. So collagen is protein. Remember. So you can what? You can recycle that into amino acids. Calcium hydroxyapatite. Once again, this is a mineral form, but certainly you can reduce it into its component elements that become the metabolites of the body. Nothing is really lost, right? Yes. Okay, so there you are. So that's endochondral ossification. All right, and as what I have mentioned a while ago, some of the bones in the skull are also cartilage. Okay, they start out as cartilage, become ossified later on. So many of the bones in the skull, especially as what I've said, on the roof of the skull are cartilage bones. Why so? We first have a cartilage model, and then after that, the bones of the roof of the skull are going to become cartilage bones. Why?
because cartilage is
No, there's no epiphysis and diaphysis in the skull bones. They are only found in the long bones. So why?
because cartilage.
This actually is going to start out as a cartilaginous piece early in embryonic development. Okay? And even at birth, you will also still retain many of these bones in cartilage form. Meaning to say there are portions of the skull when we are born which are not fully ossified. That is why you have the so-called newborn
What?
Right? Yubon.
What?
Soft areas in the soft areas of the skull that are not really holes, they are just simply cartilage which does not yet ossified. Okay? So, what is the reason for this?
Because cartilage is ideal only for womb environment, not humans.
Oh, well, okay. The womb environment is also aquatic in a similar way that we've answered the question why there are no cartilaginous tetrapods, right? Okay. But of course, the baby may be mobile inside the womb, but it's not as mobile as creatures that are free to roam the surface of the earth, isn't it? Okay? So, there must be a very good reason why our skull should be free-formed in cartilage. And why is this so?
Room to grow
This
for the
is
baby.
in relation to the fact that the nervous system undergoes very
Rapid growth.
rapid growth, especially during the first
First 3
trimester
months
of your pregnancy.
after pregnancy.
And even if the baby is born already, have you noticed that there is a ratio that favors the head part more than that of the torso or the body of the baby?
Yes.
In other words, mas taong ulo ang baby compared to the torso.
Yes.
Right? It's very unequal.
Yes.
Right? So that only presupposes that since there is rapid growth, and if the skull is not going to start out as cartilage, if it starts out as a membrane bone, there will be no room for your what? For your
Growth of the
to grow.
brain.
To grow. Right? And that would mean to say that you are going to become a moron when you go out of the womb. Correct? And of course, another reason why it has to be cartilaginous at the beginning is for the sake of the female who's going to give birth to the baby. What do you think is going to happen if the skull is fully bone when the baby is born?
It will hurt. It will hurt a lot.
Kachama ka nagadaan kung the baby's head emerges from the womb, how much more if it is bony. Correct? Alright. Have you noticed that when a baby is born, the skull is deformed?
Yes.
Kapalaba-laba siya. Right? And that is because the birth canal is elongated. Okay? But take a look at the skull of the baby as it begins to grow older. 2 months, 3 months, you will know that the head, you will observe that the head is now assuming the typical appearance of the typical shape rather of the human skull. Okay, but initially it is elongated. Okay, so there you are. So as soon as your time comes, and when is that? When you are about to deliver your baby, one of the things that you will include in your prayers, ladies, will be, thanks God for giving my baby cartilage. Alright? Otherwise, well, namaupas bilang hindi mo makalimutan. So, anyway. Okay. Alright. Because that's how you're going to feel when you deliver your baby. Alright. So, skeletal element.
Okay na.
Alright. So, we now take a look at the various components of the skeleton of the various groups of vertebrates. And when I go to this part, I can remember right away Irene's, what is this? Irene's, shall we say, thoughts here. Mas gusto niya yung anatomy lang tayo. We don't go over this. Anyway, we can escape that. So, we have dermal skeleton. And what did we say again? It's dermal bone or membrane bone. What are they? What are dermal bones or membrane bones? They are products or results of
Evolution. I'm sorry.
the results of which types of ossification? We have only two. Intramembranous, the other one is endochondral. Alright, so dermal skeleton means to say that these skeletal parts are results of intramembranous ossification. Alright, and of course, the very first vertebrates which acquired that kind of skeletal body, the very first group are those that we refer to as the ostracoderms, and that's the reason why they are called armored fishes. Shall we continue?
Yes.
Alright.
Yes.
So, where are these bony elements found?
Exoskeleton.
region. That's what are more. Alright? So, as evolution continues, or as evolution marches on, up to the tetrapods, we shall note that these dermal elements are still going to persist. And since the dermal elements were present in the head region, alright, that would mean to say that dermal elements in the tetrapods will also be seen in the head region. Okay, so where do we find dermal elements or membrane bones in the tetrapods? We find them in the skull, the jaws, and the pectoral girdle. Okay, and we will have plenty of time to enumerate them later on. But right now, here are some examples of the dermal bones that are also present in fishes. Alright? What are these? Of course. Alright, they are scales. So what do you know about scales right now? There are two things that you are going to right away consider. That scales are what? Yes, of course, they are. They may be shining, but they do not appear here, right?
Yeah.
Okay? Okay. But what do we notice about scales in relation to our previous, in relation to the previous information that we have just discussed? That they are dermal bones and therefore are what? Are intramembranously formed, correct? Yes? And so since they are formed similar to the bones of the skull and the pectoral girdle, what are they? They are going to be letter H. They are going to be H-O-M.
Hominid, homosexual.
Homologous. Okay? Okay. So there. So they are homologs. Okay. Do you know these scales are scales? And then skull bones or pectoral girdle bones are bones. But because of the manner by means of which they form, they are known as homologs. Okay? So there you are. And these are the various kinds of scales. You are going to discuss them more fully in the lab. You will be given examples of such types of scales. Right? Alright. Alright. So those were the dermal elements that were found in fishes. Now we take a look at the visceral skeleton. Right? And this visceral skeleton is now a component of what is known as the endoskeleton. For a very good reason, it's found internally. Okay? The dermal elements that were shown earlier were found outside. Now this is the endoskeleton. And one of the components of the endoskeleton is what is known as the visceral skeleton, which I think for the majority of you is something that is quite new, isn't it? Yes? But when you say visceral, visceral comes from the fact that, like when you say visceral organs, where do you find them? Inside. Alright, so visceral skeleton means to say that not only are they inside, they are also supporting structures of what should be inside the body of the organism, especially next to the head region. Right? Because we will be talking about the skull later on, separately. Okay? So, the visceral skeleton, what are they? They are associated with gills as well as the skeletal elements which are derived from them. And what are the skeletal elements that are derived from the gills? They happen to be structures of the jaw. Okay? So this statement alone already gives you a very phylogenetic information. That cartilages coming from the gills, alright, on the gill region, become the endoskeleton of the jaw. Okay? Alright, so, so here is our very generalized animal, and you will note that if this is the mouth part is actually right away here, be in the vicinity of the cartilages that are going to support the first gill slit. Okay? And what are these cartilages? They are named here, you have the palatal quadrate and the pectoral cartilage. And as of now, I would like you to say hi to those words, because they are going to be now part and parcel of our discussion. Okay? So,
Alam.
the first gill slits,
And those
alright, are supported by these two cartilages. And subsequently, the second gill slit and the rest of the other gill slits will also have their own skeletal elements. Right? And what are those skeletal elements? They are meant to support the gill slits. Okay? Alright, the second pair of cartilages that supports the second gill slit. And of course, the slit is provided with a supporting region that we refer to as the arches. Okay, a slit is a slit, right? Okay, the slit, in order to become open and closed, is going to be supported by skeletal elements. Those skeletal elements will form an arch. So that is the reason why we have gill arches and gill slits. Understood? What is the visceral skeleton that would support the gill arch? Understood? Yes. Okay. So the second gill arch, which of course is what connected to the second gill slit, is supported by the hyomandibular and the hyoid arch. Okay. Now, if the gill slits later on are not going to be needed anymore by the vertebrate because of a transformation in its lifestyle, meaning to say that organism will no longer use gill slits for respiration. What do you think will happen to the supporting elements of the gill arches? They are going to be transformed to what? To lungs. These are skeletal elements. Ribs, do you think ribs? If I were to derive my ribs, I would be deriving it from here. Alright? But these are going to disappear if I am going to become a lung-breathing organism. Alright? So I will have no need for gill slits anymore. But what do you think are going to happen to these first two pairs of cartilages? They are very near the mouth region, isn't it? Alright, so that is the reason why we said a while ago that these are going to become the jaw elements. Alright, especially your palatal quadrate and your mandibular arch. Alright, do you suppose that the hyomandibular and the hyoid arch will also undergo changes?
Yeah.
If there is a change in lifestyle?
Yes.
Yes. Will you be surprised if I'm going to say that these bones are going to become part of your middle ear bones? Is it surprising? Yes, of course, but judging from the locality or the location of these cartilage bones, you can very well see that they are going to be part of the middle ear apparatus eventually. Alright, but that is a separate, shall we say, lesson later on. Alright, now that which I really is going to love more is what we refer to as the somatic skeleton. Alright? Because what is the somatic skeleton? The somatic skeleton is not going to be the skeleton that will support the body. Like say for example, if Charles is going to stand up, what happens? So anyway, so the axial skeleton, alright, as you can see from the word axis, it supports the axis of the body. What are they known as, or what are they composed of? They are composed of the vertebral column, the ribs, the sternum, and the skull. Right? And of course, associated with the vertebral column are the limbs, correct? Alright, and what supports the limbs are what we refer to as the appendicular skeleton. Right? So when we say somatic skeleton, there are two main components. And what are those? The axial and the appendicular. Okay? So when we study the phylogeny of these structures, we will be separately studying them. So right now we are studying the vertebral column. Okay? Our next lesson is going to be on the skull, and the next lesson shall be on the appendicular skeleton, which shall be treating the ribs altogether. Okay? Alright? So for the skeletal system, we actually have 3 topics. Alright? So that will require also 3 separate quizzes. Okay? Because they are Okay. So, the endoskeleton develops from
Scleroderm.
sclerodome. Very good. You know that already. And what will happen to sclerodome is that the cells will become mesenchymatous, right? Yes? And being mesenchymatous, they have a way by means of which they can move around, correct? Yes? And where will they move? They will move around the notochord, correct? They will move around the notochord, which shall be known as the skeletogenous regions. Where else will the skeletogenous region be but around the notochord, which will be replaced later on by the vertebral column, isn't it? Okay, so there you are. The arrangement of which is largely dependent on the disposition of the myotomes. What do we mean by myotomes? Myotomes are muscle-forming mesenchyme, right? What are they again? Myotomes, dermatomes, sclerotomes, right? Yes. Okay. So, we are going to talk about this dependency of the sclerotomes on the arrangement of the myotomes later on. But right now, we are going to distinguish some terminologies. Myotomes are divided by myosepta. Alright? When you say septa, we should mean partitions. Alright? So myosepta. This is what? This is fibrous connective tissue. Yes? Alright? And this is going to divide the muscles into dorsal and ventral halves by the so-called horizontal section. So if we have here, if this is the circular view of a muscle cross-section, this is going to be divided by a myoseptum, alright, into epaxial and epaxial regions, and the myoseptum that divides them into two is going to be known as your horizontal septum. Okay? Around the notochord and the neural tube, the septum continues mid-dorsally as the dorsal septum and mid-ventrally as the ventral septum. In other words, if this is the neural tube and this is the notochord, this septum, the myoseptum, okay, are going to continue dorsally, alright, and ventrally. Okay, can you imagine? Alright, if you can imagine, then we're going to do. Okay, those are membranous elements of the myoseptum now. And down here, you also have the myoseptum to form your ventral septum. And if this is the neural tube and this is the notochord, what do you think will be surrounding this? What do you think will be found here? Do you expect to find here blood vessels and nerves?
Yes.
Definitely. Okay? Alright, but we will point them out as we continue. Alright, so, if the terms are now familiar, we can now take a look at this diagram. Okay, what is this? That's your nerve cord? What do you think? That's the notochord. That's the notochord, right? Okay. And below here, or above the notochord, is this green rod, which is your nerve cord or your spinal cord. Okay. How do you know that? Alright, because the baboshes are notochords. Okay? And what did I say a while ago? That there are blood vessels that run also the length of the body, right? Which is parallel to those of the notochord and the neural tube. So what did we say is going to happen to the sclerotomes? They're going to deposit themselves around the so-called skeletogenous regions, right? Yes? Okay. So, in connection with the skeletogenous regions, alright, we will note that the myoseptum, alright, the myoseptum is going to run this way, this way, this way, okay, which tells us that Very good. There is metamerism that is achieved simply because of the positioning of your myoceptum. Understood? Yes. Okay. And then, still another thing that you notice is that the position of the myoceptum in broken lines is also revealing of how your skeletal elements are going to form around the notochord and the neural tube. Okay, how revealing is this?
Super.
Kuda. Remember that these are skeletal regions, isn't it?
Yes.
Alright? And they are formed from skeleton simply by obeying the metameric arrangement of the parts that will develop soon. So, if I'm going to position, say for example, is kung ko ang myoceptum there? Here. Instead of here. What do you think is going to be the consequence of that? These are going to form bones. Right? These are going to form bones. The myoceptum, as you can see here, is going to be the separation or the partitioning of the muscles that are going to interact with these bony elements later on, isn't it? Alright? So if I'm going to draw this horizontally, this is how it's going to look like. If this is the sclerotum or the skeletogenous regions, and if we're going to put the myotomes over here, the okay, that would mean to say that the partitioning of the myoceptum will be such that the sclerotomes are divided in the middle, right? Yes? Okay, and that is what is shown here in this diagram. Why can't, as I have suggested a while ago, why can't we do this like this? This will be the sclerotomes, and this will be the myotomes. What do you think is going to be the disadvantage? Hindi ka. Why can't you bend? Remember, how do the muscles move the skeleton? They move by contracting. Alright, so when the muscle cells contract, they are going to become shortened, isn't it? They are going to become shortened. So if this muscle is going to become shortened later on, what will happen to the skeletal elements that are going to be, how do you refer to this? Alternately? Yes, which are alternately positioned compared to them. There is going to be what? There is going to be movement, isn't it? Okay? And of course, you understand that if this is a muscle, and this is another muscle, this will be connected to two bony elements via, can you still remember? Via its origin and insertion, right? Words still familiar?
Nope. Sorry, miss.
Wala, magid. Alright, so anyway, so origin and insertion. So what happens if, say for example, if a muscle, alright, a muscle like your deltoid is inserted into the pectoral girdle and originated in the pectoral girdle and will insert on your forearm. What will happen? A muscle. An example of that muscle is the deltoid. The deltoid is going to originate from the pectoral girdle, insert on this upper arm bone. What is the upper arm bone? The humerus. Okay? So it's inserted here. So what happens when the deltoid contracts? Okay, so it's going to pull this part of your arm towards your body, isn't it? Similarly, if you have a muscle here, what is this muscle? Your biceps. Okay? Your biceps will originate from the anterior end of your humerus, insert into the anterior end of your, what is this two? Or what are these two? The radius and the ulna. Okay? Originates on the upper section of the humerus and then insert into the anterior end of the radius. What is going to happen when the muscle contracts?
towards the
Alright,
body.
it will move towards, again, the proximal part of your body, right? Okay, so this is the reason why the arrangement must be this and not this. Okay, because every time the muscle is going to contract, again, nag-act na naman ang skeleton. Alright? Okay, what else do you notice about this diagram? What else do you notice? We have enumerated several already. You also notice that aside from the fact that the skeletonous regions are going to have separate pieces. Diba? Separate pieces? Yes?
Why?
What else does this diagram tell you? That the separate pieces will surround the what?
The neural and
Will surround the neural tube. Okay? And consequentially, if this becomes the vertebral column later on, the neural tube is going to be what?
covered in
neural tube,
bone.
alright, is going to be enveloped or covered by your skeletal pieces, isn't it? And similarly, down the line, what do we have?
Blood
You
vessels.
have also blood vessels. Of course, also the nerves correspond also to the position of the blood vessels, which will be surrounded by this separate pieces. So, if we're going to study, and by the way, the terms are there, you have the what? You have the arch, which surrounds the neural tube, that is your neural arch, okay? And then down here, you have the interhemal arch. Hemal comes from the word blood vessel, okay? And of course, interhemal arch and the hemal arch. And then, in addition to that, you will have words that you are going to meet also several times as we continue of the words pluricentrum and the intercentrum. Okay, now, if you're going to imagine the vertebra today, right now, the vertebra. How does the vertebra look like? How does the vertebra look like? You have a solid part, right? And then the solid part is going to have something here. I'm not a very good illustrator. Okay, but somehow this looks like a vertebra, isn't it? Okay, so you have a hole here. What is that hole again? That's going to be your what? Alright, that is going to be your neural arch, which will surround the what? The neural tube, otherwise your spinal cord in the adult animal. Okay? And this is supposed to be the position of the notochord, right? But what will happen to the notochord? We have mentioned several times that this is going to be squeezed out of existence, correct? In favor of the skeletal elements as shown here. So what do you think is the reason why we have words like pluricentrum and intercentrum?
It will become the center of
Why?
position.
Centrum, center, so we need to say what will happen to the skeletal pieces later on.
Matapos.
They are going to invade, yes, the notochordal tissue. Okay?
Yeah.
There you are. Separate pieces, separate sila. But as well as maturation and evolution continues, the separate pieces is going to what? They're going to fuse, they're going to merge to form one solid vertex.
Yes.
So, what do you think that we call this?
Hollow.
It's not a hollow thing. It's something that is solid. So what do we call that?
A solid.
That's what we call the centrum of the vertex.
Yes.
Okay? So you know now where the centrum arises from.
That's fine.
Okay. Now, before we go into the individual phylogeny of the vertebral column, okay? Let me ask you a question. How do we know that the separate pieces are there at the beginning? You know, our vertebral column doesn't develop like this. When we are developing inside our mother's womb, so our vertebral column is going to be a continuous structure, not separate pieces. So how do we know that the vertebral column has been designed in this manner?
Higher being.
By a higher being, yes.
Recapitulates phylogeny.
In other words, what is our evidence that indeed this is how the vertebral column first developed in the history of vertebrates?
Ancestral
Because
of a
Because
Because ontogenic
this is the condition
recapitulation.
that we see in our ancestral
Ancestral.
forms.
Ancestral.
Understood?
Yes.
Yes. Alright. So, if you're going to view and go over the phylogenetic history of the vertebral column, you will start out with separate pieces in the ancestral forms, and what happens as we progress to the more advanced vertebrates. They're going to become what? They're going to be more and more fused structures. They are no longer being solid pieces. And so what will happen to the number of terms that we're going to memorize as we progress?
Increase.
That's the beauty behind
It's so
it.
dynamic.
Okay, we start out with numerous pieces, so many terms to memorize. But as we progress to the more advanced pieces, especially like ours, there are fewer pieces to name. Okay? There.
Still too
Okay,
many.
so at least there's something positive.
It's too much
That
for my
is-
brain.
that is done. Okay. Alright, so this is what I was talking about earlier. Okay, this is the same diagram that is
Ventrolateral.
alright. So initially we were talking about what? This is going to be a very generalized diagram. Okay, but in the trunk region, the bioseptum, septum in it. Alright. This septum is divided into the ventrolateral septa by the intervention of the cilium. Remember that the cilium is the gut cavity, right? The gut cavity, which is going to expand towards the body cavity, right? So that means to say that the ventral septum is going to be divided into ventrolateral septa, and at the intersection of every myoceptum, with the dorsal, the ventral, and the horizontal septa, with the perichordal mesenchyme. What is the perichordal mesenchyme? As the name suggests, this belongs to the what? This belongs to the notochord. Okay, so at the intersection of all the septa with the perichordal mesenchyme, a vertebra arises. Okay? Alright. And as the myocepta are segmented, because the vertebra are going to intersect with this septa, so vertebrae are also segmented. Clear? Clear?
Not so crystal.
Not very clear.
So,
Alright, so this is the myoceptum, right? This is the sclerotum. So, if the myoceptum is going to intersect, alright, is going to intersect the sclerotum, which is going to be, of course, alright, because we're talking of membranous partitioning, right? So, if you have a muscle, and this is a partition, and if this is the sclerotum somewhere here, right? So, if we are going to what? If we are going to continue with this line, this is going to intersect with the sclerotum, right? So, where there is a myoceptum, there will be a vertebra. Okay? There will be a vertebra, which means to say that your sclerotum, okay, will have an upper section and a ventral section. Correct?
Yes. I guess.
Are you with me?
Yes.
Alright? That is why in this diagram here, you will notice that this is a pink region and this is a blue region. Alright?
Dorsal.
I don't know that. I will. Here. There is a pink region, there is a blue region. And what is the reason behind this? It's because this is where the myoceptum is going to intersect with the sclerotum. Right? Yes. Okay? So, eventually, what is going to happen is that the what? The anterior region is here, the posterior region is here, right? Okay? And, I don't know, okay, so what is going to happen is, this posterior region and the anterior region of the next sclerotome are going to fuse. The posterior region of this sclerotome with the anterior region of the next sclerotome are also going to fuse.
Yeah.
So that the resulting structure will be something that looks like this. Okay? No. There will be fusion of elements. Alright? Such that what is going to happen is, ini sila dua, ako na ni sila semua.
Yes.
Alright? Ini sila, ako na ni sila. Right? Okay? And the result of this is that each vertebra is going to arise where you have your what? Where you have your intersections initially was found. Correct? And where the intersection is where you are going to have the vertebra. Alright? Take note that the intersection of each succeeding vertebra will now be the form of an intervertebral disc. Correct? And that intervertebral disc are the only regions which will remind us that there was notochordal tissue before. Alright? So there you are. Okay? Now, what do you think are this?
Spinal nerve.
Alright, so there are spinal nerves. Okay? So if this is one vertebra, this is another vertebra. Okay? And the disc that will arise because of the fusions will be such that it will separate, as what I've said, the anterior and the posterior. Correct? So where will the spinal nerves emerge? In the what? In the intervertebral disc. Would you want the spinal nerves? Would you want the spinal nerves to be found in the middle of the vertebra? No, you would not want that because the vertebra is solid, isn't it? So if the spinal nerves, remember spinal nerves and blood vessels are partners, right? So if the spinal nerves as well as the blood vessels are going to arise where the intervertebral disc is found, what is going to happen is they're going to be what? This is one vertebra, it's another vertebra. So the masumot ang spinal nerve. In between. Alright? So, kung may maipit ikaw na nerve, what is going to happen? Right? You are going to pain, to be what? To be pain free? No, you are not going to be pain-free forever.
Painful.
Okay? Because this is going to be a very painful condition. Okay? Alright. So, that's the topographical relationship of your myotomes with your sclerotomes, with your spinal nerves, and the blood vessels. Alright? So earlier, what did I say? At the very beginning of this course, we said that vertebrae chordates in general show metabolism, isn't it? But this is more evident in the vertebrate formation of its structures. And this is something that we are looking at right now. So based on formation, primary metabolism is actually an attribute of skeratomes and myotomes. Understood? That's primary metabolism. Secondary metabolism, because we don't want the spinal nerves and the blood vessels to be barred, okay, from where they should grow into, alright, to achieve secondary metabolism. It's not going to be a parallel thing, okay, but they're going to develop right where it is more convenient for them to grow in. Correct? Alright? So this is why you will notice that in the vertebral column, alright, there are holes there or openings where nerves and blood vessels can penetrate through. Alright, and these are the areas or the spaces which you are going to identify in the lab. Alright, these openings become more numerous in the skull. Alright, and again, these openings and spaces are going to be identified because they have their respective names. They are referred to typically as foramina. Right, when you say foramen, you mean holes or spaces. And if you have a very big space, how would we call that?
Large.
We would call it magnum, right? That will be known as foramina magnum or the foramen magnum. And what kind of nerve will pass through the biggest foramen? The spinal cord. Okay? So, what I'm telling you right now is that there are holes and openings in our vertebral column as well as in our skull, which needs to be identified. Okay? So, take note of that. Because in the lab, you will be made to point to the foramen. Okay? When I was a student like you, I don't know if I have told you this already. I had a professor who will really put nail marks on the skull and the vertebral column. Full po kanina na is so kung may foramina na that which doesn't exist. Right? And during the lab exams, he will make you identify them. Okay? And of course, ikaw when nag-study ka na, ang tanan, and all halos memorize mo ka na nabuo sa kalibutan. Alright, but this one you cannot identify. Why? Because it's a human hole or opening in the skull. Alright? Just pray and hope that Mr. Martinez is not the same as my professor.
No.
In other words,
No,
you
it's
have